So, welcome to the Seagrass Symposium. My name is John Statton, and I'll be giving the introduction today, and there will be another 4 further speakers after me, and I'll again be talking a bit later on as well about my research, but I'm introducing the Seagrass Restoration Network, and basically what we are is a new SER community. And our aim is really to build confidence in seagrass restoration. So if anybody wants to visit the website, this is it down here, so seagrassrestoration.net, and it will give you a lot of the information that I'll talk about today, and I'll just go through a few of the little case studies that we have in there, just showcasing some of the successes that we've had across Australia. The reason why we're actually got together to put together this symposium is that last year the network, a group of core members got together and thought it'd be a good idea to start linking up a lot of the different researchers and practitioners, NGOs, consultants, and even the government people working within this space to try to build this basically a capacity or a critical capacity to actually get work done, get the outcomes that we're actually after in terms of on-ground plants in the ground and restoring these systems. So why do we need to restore seagrasses? Well, for those of you that aren't aware, across Australia we've lost close to 370,000 hectares or football field-sized areas, and this has been documented over the last 40 to 50 years. More recently, in a book chapter that a few of us here today have been involved with, we documented basically what had been happening since the last time they did an assessment or a census of seagrass loss across Australia, which was back in the late 90s, early 2000s. And there were some additional losses of seagrass around Australia, but one notable one occurred back in 2011 through a marine heat wave, which was a warming event which pushed down the Western Australian coastline, and basically Shark Bay was the epicentre of this particular warming event. The seagrass species, the 2 dominant seagrass species in this location are actually temperate species, and this is essentially a northern geographic limit. And one of these species had a massive range contraction, so there was a loss of about 860 square kilometres of this particular species in terms of its coverage in Shark Bay. So that was up to a bit over 80% of this species loss within Shark Bay. Had a massive impact on the ecosystem. There were devastating impacts to a lot of the commercial fisheries, blue manna crabs and scallops, and also the recreational fisheries, being the pink snapper fishery up there. There was also a huge input of organic matter into this system, which has some devastating effects on local industries, like the pearl farming industry just north of Monkey Mia there. The water essentially turned black, stripped all the oxygen out of the water. The oysters, pearl-producing oysters, died, and that business has since not opened its doors up since that operation. They had a large storm event as well, which influenced all the drop lines. So they haven't opened up their doors since that large event. So huge sort of ramifying impacts. In addition to this, there were some large losses or degradation in the health of dugongs and sea turtles that was also documented because of this marine heat wave. So quite a large event. This one particular event essentially doubled the amount of seagrass loss that's occurred in Australia over the past 50 years. So it was quite a huge event. In total, collectively across Australia, we've probably actively restored less than 50 hectares of seagrass since the 1980s. So you can see there's a huge difference in these numbers here, and basically we're getting together as a network to try and work out how to get things done. So the core team got together in Geelong in 2017. There were 16 members initially, sat down and workshopped a few ideas about ways forward, and this particular network and the website that went along with that was one of these ideas. Since then, we've grown to 59 members, and these members come from a whole range of different areas. So there's lots of universities across the country with active seagrass conservation and restoration members in those universities, members from Nature Conservancy, CSIRO, councils, NGOs, private sectors, and other government as well. So I won't list all those, but you can see that just in a short space of time, we've gathered quite a bit of interest across the country, and also not just within the Australasian region, but also in the US and parts of Europe as well. So there's a lot of people taking interest in what we're doing, and some of the success stories that are actually coming out of it. So the mission of the Seagrass Restoration Network, I'm just going to read this verbatim, was to enable the sharing of knowledge and tools for seagrass conservation, recovery and restoration, and foster an integrated long-term approach to developing restoration solutions. And so I think that's really key for us, is really sharing of knowledge and tools. That's why the network got together. It was essentially an open forum where we could openly discuss, free from reprimand, the research that we're doing, ways in which we can collaborate, ways in which we can basically join forces to be a larger front when it comes to trying to return seagrasses back into the ocean. The Seagrass Restoration Network Australasia aims to link the scientists, the industry practitioners, communities, NGOs, government policymakers, and as you saw before from our member list, you can see we're already on the way to bringing those different groups together in a number of different capacities. Just to give you a couple of quick examples of how some very recent programs, one of which will be talked about today, which is Operation Posidonia. These particular programs have brought together government agencies, university and environmental trusts and different sort of community sectors and community groups, which is what Lana will be talking about pretty heavily about, together in this particular program here. And it's been a very rapid progress, I think, essentially. So once this program started up, we were pretty much on the ground as soon as the funding came in. And this is really starting to bring together a lot of people. And this research is being undertaken in Port Stephens in New South Wales, and it is looking at basically using a citizen science program to try to bring back seagrasses within moorings, in particular environmentally friendly moorings, but that will be talked about in detail later on. This particular program is one that's just started up in Western Australia. It's using a not-for-profit group called Ozfish Unlimited, which are basically a group which come together to restore habitats to improve fisheries. So they'll be putting snags back in waterways for habitat for fish. They'll be, be involved in oyster reef habitats as well. And they saw seagrasses and the research that we're doing, which I'll be presenting a little bit later on, as really that step for them to go from the eastern states of Australia over to the western states and use this particular program. And they're using the pink snapper here to really engage the fishing industry, the recreational fishing industry. And so we're getting not only recreational fishermen, but also the peak recreational fishing body in Western Australia, which is Recfish West, involved in this program. So we're already starting to have a bit of reach with regards to what this network is actually capable of. So the breadth of research, I suppose, from the academic perspective, really bridges a lot of the gaps across the biological scales, from genetics, populations, communities, ecosystems, all the way down to sort of climate change effects. So the expertise that we have in this network is quite broad. And this is just to give you some examples of this network. So we have population geneticists. So a lot of this research that we've been doing, both on the western and eastern coast, is really trying to understand the connectivity amongst different seagrass populations and provenance issues with regards to sourcing plant populations for restoration. Hydrodynamic modelling, so looking at where propagules can be dispersed to, for example, where fruit carrying seeds get dispersed to, or where spathes or fragments of seagrass can potentially be dispersed to. So essentially areas that are sinks or natural sinks for seeds to get to. We've also, more recently, probably engaging more heavily in social ecology, so people looking at quantifying the impact that our research and our programs are having on the community and how we're engaged in that community and if that's actually having an impact on the outcomes of restoration as well. And also, we're looking at doing some cost-benefit analysis of our restoration programs nationally as well. So we're engaging economists in our work. But where we don't have the expertise, we go ahead and try and engage external expertise. So just quickly going through a few case studies, some success stories, which is really what a lot of people have been coming to the website for. This is in New Zealand. I have no idea how to pronounce that. Thank you very much. You're correct, Lana. So in this place, excuse my ignorance, so Fleur Matheson has been working at using different transplanting techniques to bring back Zostrum muelleri to the intertidal area adjacent to this harbour. And so essentially across this whole area, the seagrasses historically covered about 10% of the coverage in the intertidal area. What she's been able to do through using either sods, which are large transplants, or sprigs, which are just the individual cuttings of the plants, she's been translocating these from remnant areas, and she's been able to expand it from, it was basically had about 10% coverage in the past, and she's been able to expand that over only a short space of time, 2 to 3 years, to now covering 40% of its former range. And so basically by putting out these plants, they've essentially ramified across that area. It's a very fast-growing species, and has been able to cover several hectares in the process, which is an incredibly successful restoration story, which seems to be quite new. I've only just seen this come up, so it's great. It's quite great. Another case study is in Western Australia and Oyster Harbour, so Jeff Bastian there in Cambridge. Jeff Bastian, for his work in trying to restore Posidonia australis in Oyster Harbour, won an award, the Southern Oceans Hero Award, followed by a medal, the Southern Development Commission medal in 2016 for his work. So he single-handedly went out there and planted out thousands and thousands of plants of Posidonia australis. So here, he started the work back in 1994. Here in 2001, you can see these patches now. They were 10 by 10 metre areas, and each one of these little dots is an individual plant which is essentially grown and ramified out. And so it's starting to come up in aerial imagery. These are naturally recruited patches. That's why they're sort of these concentric circles. And then 10 years later, you can see these squares here, these 10 by 10 metre areas have completely infilled and are basically creating these small patches, essentially. And these have become, since then, have become reproductive and spread on and started producing seeds. Naturally, and you can see these other little dots. So this is all natural recruitment from seed set that has then expanded. So another great success story there, using transplanting of adult plants. Another great story, and one that uses a very different technique, is in the Coorong. Basically, long-term drought events had really reduced water flow into this particular area, and with the loss of the main seagrass there, Rupia tuberosa. And so what they did was, there were still seeds sitting in the sediments, and those seeds were still viable. So what they did is they used a technique which is used quite commonly in terrestrial systems. They took that topsoil of seed, with seed in it, put it into bags, and they took those bags out across your tidal area and put these, and basically empty these bags. And then over a very short space of time, the seeds inside these bags germinated and these small patches grew. And they were actually able to create near complete meadows through this process. They transferred about 700 tonnes of topsoil and covered about 61 hectares of area. Great success story. The unfortunate problem is that because of the catchment issue still going on in that location, a lot of those seagrasses died off. So the technique's there and it works great, but there are other issues sort of further upstream which are really causing problems, and these are things like eutrophication and water flow in the system. So I'm about to finish up, I'm almost at 15 minutes. So today there'll be 5 presenters that will be talking today, and we've brought these people together because they've got a diverse range of expertise. And we're seeing this today as really a sharing of knowledge and tools that have been going across a wide geographic region from both temperate and tropical, west and east side of the country. People bring together to this symposium different practical techniques and novel approaches to their research, and also innovation and social network analysis will be brought into this. So it's really good having a social scientist brought into the symposium. It's added a different flavour. And also there'll be a couple of presentations on seed and seedling ecology. Thank you very much. Okay. So, would Perfect. So our first presenter for today will be Emma Jackson. And I'll leave it up to you to introduce it.
Thank you. Oh yeah, not very good. Probably scary. Thank you. Hello everybody, thanks for coming along. So I'm going to talk a little bit about some of the seagrass seed work that we've been doing, but the first thing I wanted to do is put the record straight a little bit. So yesterday at SERA Torres's plenary talk, she talked about coral restoration as being the Jon Snow of ecological restoration. Okay. Yesterday, John basically called seagrass, which I was a bit upset by, the ugly cousin of coral reefs. So I did a little bit of research, and I found, well, if Daenerys Targaryen is Jon Snow's auntie, that means that seagrasses are actually the dragons of ecological restoration, because she's the mother of dragons. So I think that sounds a little bit better. And sea dragons are really also one of the species that's actually supported by seagrasses. And I don't know if you noticed with John's talk that the logo for our Seagrass Restoration Network doesn't have a picture of seagrass in it. And the reason we decided to go for this is just to show people that aren't familiar with seagrasses just quite how much they are a habitat for a lot of iconic species and fishery species, and really get people to engage with the fact that these seagrasses are habitats. So you can see the sea dragon in there. Actually, there's a leafy sea dragon as well, so two dragons. So it's the right logo. And of course, we've seen over, and we'll see, I'm sure, in the next few talks, all the other important ecosystem services that seagrasses deliver. And I won't go through them for time. And yesterday, John spoke about the fact that we're actually losing these at an increasing rate. So 25% loss globally. We saw from John's talk just then that Australia is probably a lot higher than that in terms of percentage losses. And this rate is increasing, and it's increasing at a rate that's a lot faster than the natural recovery of these species. A global meta-analysis, a review in 2016, actually found that under trials, the survival rate of trials for seagrass restoration are about 37%. But this was a great study because it actually pulled out all of the reasons for those failures and used those failures and successes to come up with a number of different things that we can think about to improve things. The biggest one, and it's been mentioned a lot over the course of this and other conferences, is this scaling up idea. You know, plant more, plant a larger area, get those sort of self-sustaining feedbacks back, cover lots of environmental variability so that you've got more chance of success. So a numbers game of actually putting things out there. But the other thing that it highlighted was that idea that you, the more you learn about a system, the more you understand your system, the more you understand seagrasses and the methods and techniques that are involved, and have that built up over a number of years, the more likely you are to succeed with your seagrass restoration. And I think for people that are just starting out in certain areas for seagrass restoration, that's where the Seagrass Network is going to become really, really important, because you're relying on other people's experience as well, and you're able to bring all that together. So we're working in central Queensland, just here, in a place called Gladstone, which some of you might have heard of. So Gladstone is like the fourth largest coal port in the world. It's Queensland's largest multi-commodity port, and there's a lot going on there. We've basically recently just had three, not one, but three liquefied natural gas plants built on the coast here, and there's everything from coal to smelters and cement works, and you name it, Gladstone's got it. They're also talking about extending dredge channels to actually allow bigger and more ships to actually come in and to be able to pass each other. So it's a continuously developing port. The seagrass there has basically been monitored for a number of years, so we've got really good indication of how much loss and degradation we've got in the harbour. So this is Rob Coles' paper, and you can see, so this is Gladstone in the middle here. And there's been a 50 to 75% loss in seagrass coverage over the last few decades. And primarily this is due to poor catchment management and lots of sediments and things coming down from the catchment and nutrients. But also, and this is a big one, the actual land reclamation and the place to put all that dredge spoil that they get from all the capital dredging, basically gets put into these big bunds and covering up a lot of seagrass habitat. I'd like to say this is Gladstone, but it's not. This is where I used to work. Same genus, Zostra marina, but now I work on Zostra muelleri, and in Gladstone, I saw Fleur's picture in your talk, John, but in Gladstone it looks a little bit more like this, so I'm not quite sure why I moved, but anyway. So yeah, it's very muddy. The seagrass is very short, it's very, very heavily grazed, and it's in some areas really quite sparse, so a very, very different system. And unlike some of the other talks that you're going to hear about, the seagrasses there tend to be in the sort of like colonising and opportunistic sort of end of the scale, rather than these sort of persistent meadows like the Posidonia and the Amphibolis that you might hear about. So this means that they are fast growing, which is good, but they're also very prone to being disturbed, and you tend to basically have a lot of difficulties in actually making a persistent meadow. And not just the form of the actual species being different and being these colonising opportunistic, but because we've got a lot of events in the subtropics and the tropics that actually cause these massive disturbances, they're continuously being disturbed. So you actually have a higher proportion of these transitory meadows because of these opportunistic and colonising species not being in the very sort of like stable conditions. So what we end up with in Gladstone is actually a meta-population of different meadows. Some are transitory, some are persistent, and some areas of suitable habitat might be colonised by seagrasses and some might not at any one time. And the ones that are colonised might be connected at different extents. So some of them might be sources of propagules for other meadows, and some of them might be sinks of propagules for other meadows. So it's a real classic sort of meta-population type system that we've got to get a handle of. And we've also got to, it's important to understand this because it means that, one, your seagrasses might, that you restore, might disappear after a few years, and you've got to just like accept that. It also means that if we lose too many of these source meadows, we might actually sort of see a population collapse in the area. It also means that there are areas that are suitable habitat and propagules aren't getting there. So these are really prime places to go in and put propagules in to actually restore. But there's also areas where the propagules are going, but the habitat isn't suitable, so there's potential for doing habitat enhancement for these sites as well. But the big thing I think that this tells us is that the restoration in these particular environments is not going to be a one-off thing. You don't go to an area that's lost seagrass, the conditions are good again, you put seagrass back, you walk away, you leave it alone. It's going to be a continuous program of thinking where the seagrass needs to be enhanced at particular times going forwards. So really, it's not about just spatial scaling up of restoration, it's that temporal scaling up of restoration and seeing it as basically being as common as monitoring. Now with this scaling up, we've done a little bit of work looking at the sods and the sort of things that John was describing Fleur was doing in New Zealand and had a bit of success with that. But with the scaling up, it really means starting to look at seed work. For a number of reasons. You can scale up with seeds without having as much donor bed damage. You're not reducing genetic diversity as much, and obviously it's not costing as much to use seeds, we hope. But we don't know what the relative success of this is. So there's a lot already known about seagrass seeds and how we might use them for restoration, but we're still a long way from actually getting the sort of information that we take for granted on the back of a seed packet. So we need to we know things like the germination rates increase with decreasing salinity, that there's variations in temperature that influence the salinity and germination. We know that darkness can promote hypocotyl hairs. We know all these little bits of information. But what we found is that they're very specific to where that seagrass was from, and it's very specific to the local environment as well. So what we're trying to do is build up this sort of information for seagrasses in our local area. So everything from what density to put the seeds out, what burial depth to use, what's the sell-by date of the seeds, you know, how long can we actually keep the seeds and keep them viable, all that information that there's been some really great terrestrial talks on at this conference. I've been scribbling madly and learning a lot. And like I said, the terrestrial environments, this is sort of road mapped out. So we're not just like picking things to look at and just randomly doing it. We're thinking, right, what do we need to actually build up in terms of information? But it is a massive task to get it right. And we saw that great talk this morning about the genetics and looking at seeds and the genetics of the seeds. And you know, that's just one small bit of it. And all the rest of it has to be built up as well. Oh, get overwhelmed when I think about it. But part of a net right now, it's fine. the little bit that we're focusing on at the moment is looking at the variability in flowering, so that we can try and predict when and where the seagrass is going to be flowering, because it's incredibly patchy, even in our small area of the world. Looking at what affects the germination rates and what affects them naturally, and how we can use this to actually look at germination and enhance germination. And then looking at some of the factors that might influence these tiny little seedlings that are formed, and then they get hit by massive flood events and cyclones when they've only just formed. So getting over some of these hurdles is going to be really important. So like I said, our seagrass isn't this long, lush seagrass that John showed in his talk yesterday in VIMS, where they basically take this sled and they drag it over to collect the flowering plants, and then they basically take those, put them in their sort of like big aquarium system and let the seeds drop out. We can't use any sort of like mechanical method at the moment, which doesn't basically totally trash the seagrass, because the seagrass flowers are just sticking out from the bottom of the sediment. They're really close to the ground. But we've got a really good sort of like volunteer and citizen science program now, where we get volunteers from the local community, from schools, also from the local indigenous sea rangers that come along and help us just collect flowers en masse. And then again, we do the same sort of thing. We basically put them out into aquaria systems, let the seeds drop out, and then we can collect the seeds that way and use them in our experiments. And we're just going to finish off by talking a little bit about a couple of the experiments that we've been doing. There's a whole range, too many to like go through in this, but just a couple of them, just to give you an idea of the sort of things, the little individual experiments that we're having to like build up. So the first thing that we looked at was, what are the best conditions for germination, and are they the same across all sites? So we knew already that freshwater pulses can stimulate germination, but we wanted to know basically how fresh can it go, and how much of a duration of fresh water, bearing in mind what they're actually exposed to out in the environment. If we were to put ungerminated seeds out in the environment, would they actually get that level of exposure to fresh water? So we exposed them to 2 different salinities at for different durations in the light and the dark, and had a look at the germination rates after 80 days. And we found that there was a slight effect of sight, and we're looking into this a little bit more. But the big thing was that the salinity came out as being significantly different as a main effect. But at 2 fresher sort of temperatures, so temperature salinities, then the germination rate was compromised a little bit, and you actually got better germination rates when you went up to a higher salinity, which is what they're exposed to most likely out in the field. And this got us thinking about the other sort of natural processes of dispersal and settlement that might influence germination rates. So the seagrass seeds can either drop out onto the sediment, they could pass through the gut of a dugong or a turtle, they can be in little spades and float on the surface, like hundreds of kilometres up the coast in really sort of like high sunlight and really high salinities, or they can get buried and moved along in the sediment. So we looked at the different ways that these could actually be transported, and recreated some of these environments. And what we found was that passing it through what was our sort of pretend gut of a dugong, and also those sort of like strong, very different conditions of being exposed to the light, actually gave the best germination rates. And we're going to look at this in a little bit more detail this year. We also looked at seed burial and sediment composition, and looked at how that influenced seedling early survival. And we found, so this time we used pre-germinated seeds, that we know that they're viable and know that they've germinated, and we buried them in different types of sediment and also in different depths of sediment. And what we found was that we didn't find any effect of the type of sediment, but what we found was that if they were just put on the surface, you got a lower seedling emergence than if you actually buried them at 5 millimetres depth. But then at 10 millimetres depth, they were too deep. And I saw an interesting talk, I think it was on Tuesday, where they actually related seed size to that maximum depth of emergence, and it fits really nicely with that. So they are too deep. So this really means that, and what happened here was that as soon as that first leaf came out, they started to float off and disappear, which is what would happen in the natural environment. So, okay, finished now. Right. So looking into ways of combating this, because it's going to be very difficult to bury them all at 5 millimetres, so seed coating and things like that. last slide. So seeds are definitely a more practical option. The important thing in our area is that it's not going to be a one-off restoration effort, and we also need to look at practical ways that we can get these seeds out there, because burial is going to be a bit of an issue. But the key thing is going to be part of this network and sharing the knowledge. Thank you. Sorry.
Thank you, Emma. I think we might keep moving on.
Sorry.
Our next speaker is Jason Tanner. He's from Statton in Adelaide, and he's going to talk to us about the large temperate seagrass species and the restoration of that in Adelaide waters.
Thanks, Liz. So yeah, I'll be talking about rehabilitation program we've got on Amphibolis Antarctica. It's a species we started working on in 2004 or something like that, so we've been working on it for quite a while and starting to have some reasonable success now. So we're in Adelaide here in South Australia. We've lost about 6,000 hectares of seagrass off the metropolitan coast since 1949, mostly in shallow waters rather than deep. And that's primarily been due to eutrophication and turbidity from wastewater treatment plants and stormwater inputs. The main taxa that have been lost have been Posidonia and Amphibolis, so the big structural forming species. And there's been a lot of work put in over the last decade or so to clean up some of those inputs. We've had a major industry shut down that was putting in 1,000 tonnes of nitrogen to the system. SA Water have cleaned up a lot of their wastewater treatment plant inputs, and so nutrient levels are probably, or nutrient inputs are probably 25% of what they were at their peak. And that sort of suggested to us that it was time to start looking at promoting restoration in the area. And this is just a map of some of our seagrass loss, in particular yellow and red areas is where we've lost a lot of our seagrass, and that's our main study site there. So I thought I'd just start with a video showing the natural seagrass adjacent to our study site. So fairly dense, still fairly heavily epiphytised, and then a very, very clear demarcation between the seagrass and the area of loss. And one thing you'll notice here is all these mounds, so very high level of bioturbation, as well as any decent storm will stir up all that sediment. And that's why we're not getting natural recruitment. Anything that comes back in here and starts to get established, the next winter storm, it'll get ripped out if it hasn't already been buried by all these shrimps and worms that are causing this bioturbation. And so we had to come up with a technique that actually overcome those issues. So when the program was first started, all the traditional techniques of transplantation of cores and sprigs and germinating seedlings in tanks and planting them out were tried, but we hit on this technique of recruitment facilitation. So the Amphibolis, the main species we're targeting, is actually viviparous. It releases these little seedlings about yay big in July of every year, and they have this little cone-like apparatus on the end of them that acts like a grappling hook. So it looks like they've evolved for the seedlings to actually entangle in exposed root matter, things like Posidonia. And quite fortuitously, Hessian sandbags act as a mimic for that exposed root mat. And so what we're using is these sandbags to actually promote natural recruitment. So we just put them out. Here they're all nicely laid out from our experiment, so we can go back and follow them over time. Operationally, we just toss them over the side of a boat and leave them, which makes it a lot easier than any planting or anything like that. So here's that same area of rehabilitation a year later. You can see each sandbag has got hundreds of these recruits on it. So they're just naturally drifting past in the water column, and they tangle up on the Hessian. You'll also notice here that the bags themselves have all been covered by sand, so you've got to be careful about your timing. If you put them out too early, they'll be covered in sand and the recruits won't attach. So we were getting upwards of 200 recruits per bag early on when they were first put out. This is compared to a natural stem density in the adjacent meadow of about 20 stems per in an area the equivalent of a bag. So that looked quite exciting. And then sort of a year later, some of those bags we were getting a second peak on. Still looking reasonably good, but we were getting a bit worried here when we were losing quite a few of those early recruits. And a few more years later, we were sort of thinking, oh no, this is a failure. Not much is happening. We've lost most of the stems. And it continued like that for a few years. And one of the major issues we've had, I guess more recently, is that some of the managers have sort of seen this data and completely given up, and we're now really struggling to convince them that, well, those early results don't actually tell you what the long-term success is. So as I said, yeah, it continued that way for a number of years, but in the last couple of years, those plots are now suddenly shooting up in stem densities. And the managers are sort of just keeping on ignoring that and saying, no, look at this data, it's all failed. So that's a really big blocking point that we've had at recent. If we look at the actual stem length, again, here's the mean stem length in the natural meadows, about 30 centimetres. We're getting up to that after about 4 years, and in some of them, plots actually exceeding that before they're starting to drop back down. So here's a video of one of our restoration plots. If you remember back to the first video, it looks very similar. So high densities, we've got fish and various other things recruiting in. So it's looking now like a natural seagrass meadow. And still these areas in between our patches have got absolutely nothing in them. And what we'll see coming up just in the top corner here is that we're also starting to get some recruitment of Posidonia just here, actually recruiting now naturally around the edges of our rehabilitation plots. So while they're primarily targeting Amphibolis, once you get the Amphibolis in place, then the other species come in as well. So as well as looking like natural meadows or natural patches of meadows, I had a PhD student, Chloe McSkimming, starting to look at some of the ecological processes occurring in those restoration patches. And this is just one brief graph from her work looking at faunal abundance. And we can see in our restoration plots here in the white have basically equivalent faunal abundance as what we had on both the edge and the interior of the adjacent natural meadow. If we looked at faunal composition, again the same thing. Within about 5 years, the actual composition of the fauna looks the same. And by fauna, I'm talking about all the sort of little amphipods and other crustaceans and molluscs and that sort of thing. These patches aren't yet big enough to start sampling fish in, so we don't know if we've got natural fish abundances. So that was at a small spatial scale. Our plot that we'd used over a number of years was probably no bigger than this lecture theatre. We then decided to scale up to a 1 hectare trial. And at first glance, this looked like a failure. This is stem densities after 6 months in the purple in all our small-scale plots. So emphasising here that over winter is the successful ones. Plots put out over summer didn't do too well. And our large-scale plot pretty much looked like a plot put out over summer. So we sort of thought that was a bit of a failure. But we did go back about a year and a half later and things started to look a bit better. Relatively speaking, the stem density had actually increased compared to most of our small-scale plots. Unfortunately, after this, we were exposed to a major disturbance. I suspect by fishing, because because all our marker stakes have been pulled out, and that also damaged a lot of the seagrasses in the plot. Now, one of the issues we had with these 1 hectare scale plots is that our bag densities were very low. We put out 1,000 bags in 1 hectare, which is 1 bag per 10 square metres. Our small scale trials were about 1 bag per square metre. So I think that also had a major role to play in how successful this plot was. So the next step now with the Amphibolis is that we've just got new funding to actually do what we're calling a proof of application. So we're going in at that 1 hectare scale, going to put in multiple plots with different bag densities, up to 10,000 bags per hectare, which will be the equivalent bag density to our small scale plots. So hopefully with that we'll actually get successful 1 hectare scale restoration. And by using different densities, we'll be able to establish what density we actually need to operationalise this particular technique. So that's the work we've been doing on Amphibolis. We've also been playing around a little bit with Posidonia, mostly on the side, looking at using seeds and seedlings. So we've done a number of different trials, both in situ and in the lab, looking at different sand mixes in the bags and in pots to see how they respond. So again, we use the same Hessian sandbags. This time, unfortunately, we can't rely on natural recruitment. We've got to go down with divers and plant them in. But at least all this data I'm showing is with divers. My latest experiment has shown that we don't actually need to use divers. We can plant them into the bags before we drop them over the side, and they survive just as well. And so again, the same layout, but this time the bags have got different sediment compositions in them. And here's just one example where we've used different mixes of sand and clay, followed them over time. You find here that a 50-50 mix of sand and clay seem to be the most successful in the long term. You can see here we've also got high leaf counts, then sudden declines over winter, probably related to winter storms, then increased. And then again we had the same problem as we had with the 1 hectare trial. All our marker stakes got ripped out and the actual plots seem to get decimated. So again, we've got this major disturbance happening that we've got to pin down what it is before we go too much further. So here's just an example of one of our bags a couple of years after we'd planted it out. By this point, we couldn't follow individual plants. They just started to send out rhizomes and shoots throughout the whole bag. And that's sort of a broad scale plot, again, after a couple of years, showing all our different bags with the seedlings sort of becoming established and becoming mature. So next step, as I said, is scaling up to multiple 1 hectare scale trials, but we've also got money to do some reef rehabilitation. So we're starting to think about landscape restoration and mosaics of reef and seagrass rather than just seagrass restoration over there and reef restoration over there and sort of ignoring the interplay between the two. So we're interested in to see if they can actually promote each other and we get synergies between the two. So I'll leave it there. Thanks very much.
A couple of minutes. Does anybody have any questions for Jason before we move on to the next speaker? Any questions?
No, there's about 20 kilos each. So once we drop them there, they'll stay. As I said, they actually fairly quickly get buried under a bit of sand, so that helps. And then after a year or two, they start to disintegrate, which is actually what we want. We don't want something that's going to be there still in 20 years' time. We want it there just long enough for these seedlings to get themselves established so they can resist that sand movement and then for it to biodegrade.
Has the sandbag technique lend itself
So I don't think it's too useful for citizen scientists here because we're in 7 to 8 metres of water, and most citizens have relatively small boats, and so you're going to have stability issues and that sort of thing. Even in our big 8-metre boats, we can only carry about 10 or 15 sandbags at once. If we want to put out multiple bags, we need to use our 25-metre vessel for that. But potentially, if you've got areas where sediment disturbance is a problem in shallow waters, then it might be useful in that situation. Yes, I think the eutrophication is, it's not totally eliminated, but it's certainly a lot better. And then we've got basically that hysteresis effect where you've lost the seagrass, that means the sediments become mobile and the seagrass can't come back by itself. And so that's what the bags are doing, is overcoming that hysteresis.
Great, thank you very much, Jason, for that presentation. We'll move on to the next. Okay, so up next is Elizabeth Sinclair, and she'll be talking to us about the project I mentioned before, so the Operation Posidonia. Yeah, so I'll let you do it. Okay. All yours.
Okay, so I'm going to talk to you about boat moorings and the impact they have on seagrasses. So I think we're all well aware, if you're not until you arrived here, you are certainly now the huge impact that humans are having on the coastal marine environment. We've got a huge population living on the coast, and that's only set to grow, which is going to cause more and more stress on our environments through the impact of coastal infrastructure. And some of these examples here you might well be familiar with. Up in the top corner here, this is actually the planned expansion in Coburn Sound in Western Australia for our new port. So it's going to have a huge impact along the coast here. We all know about what goes on in Caribbean resorts and how they remove seagrass to build these lovely resorts. And of course, island building structures that go on in the Middle East and also the South China Sea, you would have heard as well. So the main reason for this conflict in terms of boat moorings is that we love to park our boats on these nice sheltered embayments along the coast, and that's where the seagrass meadows really thrive. So trying to find a way that can both coexist is really important. There's obviously major problems in Europe, in the Mediterranean. We have them here in Australia and also in the US. I think the awareness is gradually building, and just a quick trawl through the literature found these 4 papers that are talking about the problem and trying to quantify the problem and look at possible solutions. So just briefly, the problem occurs when people use block and chain moorings, and these large heavy chains drag around the seagrass meadow when the wind changes, and initially they just take the leaf material off. But the longer the mooring is there and the more movement removes the large rhizome material that holds the sediment together. And once that occurs, it's very difficult to regrow seagrass. Whereas if we use one of the environmentally friendly options which are now available, they keep the chain material up off the seagrass, so you can actually grow the seagrass right in close to the concrete block, losing a lot less seagrass. So an individual mooring scar is quite small in terms of footprint, but the problem exists when we have a lot of moorings together, a lot close together, and these scars gradually expand and join together, causing losses of whole meadows. So as I said, the rhizome material where shoot growth occurs from is eroded away, and then it's really impossible for the seagrass to regenerate itself. So it becomes this self-perpetuating process which really gets on the wrong trajectory. So I'm going to focus in on New South Wales. Up until the 1980s, between about 12 up to about 57% of seagrass has been lost in individual estuaries along the New South Wales coast. And so in 2010, New South Wales Fisheries put in place some legislation whereby the seagrass meadows that occur between Lake Macquarie and Port Hacking are now listed as endangered. And this includes 6 estuaries and includes the large around Sydney, Botany Bay and so on, where we've got large human populations, and they've had a huge impact on the Posidonia seagrass meadows. Further, in 2015, the federal government actually enacted their threatened ecological communities and designated this whole chunk of coastline, which includes the Manning-Hawkesbury region, and that includes just about all of the seagrass meadows, Posidonia seagrass meadows in New South Wales. So they do have a lot of protection now, but what a recent study by one of my PhD students showed was that we are still seeing significant declines in the seagrass meadows, and particularly in these endangered systems. So the red here shows declines in 5 of the estuaries, which are 5 of the 6 endangered estuaries. And we see this incredibly large losses still going on in Sydney Harbour. And they're occurring at a rate of about 10% a year, which is higher than the global average loss for seagrass. And I think this photo really shows what's going on. This is a single mooring in Manly Cove in the eastern suburbs of Sydney. This was a single boat which had an EFM, so an environmentally friendly mooring. But for some reason the mooring was changed to a block and chain mooring, and within 2 years we can see this large scar already opening up within a fragmented meadow. And if we actually zoom out and look at the whole of Manly Cove, this is 2 shots taken 5 years apart, we can see that this meadow is just continuing to decline. The boat mooring scars are connecting, we're losing the meadow, and that's about 95% loss in that meadow. So certainly there's a lot of challenges to trying to do restoration in New South Wales. 11 of the 14 estuaries are fairly heavily impacted in terms of loss of Posidonia meadow. These declines are continuing. We've got 19,000 private moorings, it's estimated, across New South Wales. So that's a lot of holes in seagrass meadow. And the majority of these are for private boat use and they're block and chain moorings. So the seagrass decline will continue. It's estimated about 9.5 hectares of loss so far. So one of the challenges is that there's been a really low uptake of environmental friendly moorings, and until those moorings are changed, there's really no point in trying to restore these scars. And it's come about, I think, in part because of poor perception of there's been poor restoration success in New South Wales, and this perception continues despite the fact that we have had some wins in terms of seagrass restoration on the scale that we need to start to repair some of this damage. An earlier genetics study that I was involved in, we found that each of the estuaries are fairly genetically distinct meadows, and so there's not a lot of connectivity now between these estuaries, which makes sense. There's low seed production, so there's no genetic connectivity movement of seeds between estuaries. And a recent restoration trial we conducted also showed that there's a fairly strong tendency towards home site advantage. So those seagrass seeds or transplants that were put back into their home site did a lot better than those that were brought in from different sites. So these 2 facts alone start to limit the amount of material that you have available to do restoration. And then when we add on the fact that this species is now listed as a threatened ecological community, we cannot go and harvest rhizome material to do transplant programs and start to recover the seagrass. So we had to get a little bit creative here. So this is our new project. It's just been funded by the Environment Trust in New South Wales. It's called Operation Posidonia. And what we're trying to do is use 3 different strategies to change people's ideas, try to prove that we can restore these moorings, and then try to improve community awareness and sort of expand our scale at which we're going to do some restoration. So our trial site is up in Port Stephens. Our collaborators are there with DPI, so it makes it logistically much easier to work with. We have tanks available to us to store our rhizome material in for transplant experiments. So, and Port Stephens has lost about 2 hectares of seagrass to mooring scars. The meadows are largely Posidonia australis, but some of the mooring scars have now started to recover on their own, but using smaller colonising species. So late last year, and our way of collecting material is going to be through collecting rhizome material that's washed up on beaches. This is material that's already been dislodged from seagrass meadows, and it gets washed up after storms. So this material is essentially dead from the environment, from the system until we put it back in. So this gives us free plant material to go and recover and put back into the seagrass mooring scars. So there's 2 collections conducted, one late last year and then one earlier this year. And in May, all of our research team went to Port Stephens and set up a trial just to test a few different techniques, see what we've learnt from successful restoration in Western Australia, and learn a little bit more about Port Stephens and how we might need to adapt our protocols to make it successful. So the seagrass that was collected, washed up on the beach, is just kept in these holding tanks. There's small trays here with about 50 millimetres of sand, and the rhizome is just planted into that, and that's essentially used to keep it alive until we can go out and plant material. This is Shoal Bay in Port Stephens, and we can see here the moorings that exist, and we have the standard block and chain moorings, which are fairly common throughout the bay. But we also have some environmentally friendly moorings which have been installed. They're also quite close to some of the block and chain moorings. So for our trial, what we actually decided to do was come up to the side here where there are large mooring scars, but there are no boat moorings at the moment. We wanted to just make sure that we could grow the plants first rather than grow them under potential conditions where their survival would be compromised. So we've used 2 sets of mooring scars. Some are completely bare sand, as you can see here, and other ones have got Holophila and Zostra starting to move in. And so these are they're small colonising species, but we thought maybe if they'd already crept in, then the sediment might be a little bit more stable and be able to permit the growth of Posidonia in there as well. So we've put transplants in under both conditions. You can see the Posidonia here. And they were just planted straight into bare sand. And then the rhizome is held in place by basically a piece of wire peg. In some cases, the rhizome was pretty small, and so there's also a little attachment to hold the rhizome to the peg. In the other set of conditions, and this was only done on bare sand, we've used hessian or jute matting, similar to what Jason was talking about. But rather than create sandbags, the jute matting is attached to rebar on both ends. And so we have about 1 metre by 1.5 metres rectangular piece of jute matting, which we put on the seafloor, make sure it's fairly firm, and then we actually plant straight into that. So in this case, we planted the rhizome, just wove it into the jute matting to avoid pegging it, and then in this other site here, we've actually put the pegs down to hold the rhizome in place, and just to see which method worked better. What we're trying to do is find a really easy, quick planting method so that we can actually spend less time on one site and actually, scale this up a bit better. So we've just got some really early results here. We checked survival at 48 days, and then again just last week after 125 days. And there's some promising results here. We seem to have much better survival going on in the bare sand sites than we do in the Zostra Holophila colonising species, which is great because it's much easier to set up on bare sand. We also see that there's survival of the shoots that have been kept for a lot shorter period of time in tanks. So those plants that were kept in tanks and had been there for about 5 months, they'd grown really nice roots, and we had to pull them up and then replant them. But those, I think, so much energy went into growing those roots that they're not going to be able to regrow another lot of roots and attach to the sediment. So their survival is much lower. So the less time we hold plants in the holding tanks, the better. And also the rhizome really needs to be well secured to the sediment for it to start growing properly. So holding them, they seem to do better. And again here, these were pegged in and they do a bit better as well. So that's giving us a pretty good idea of how things are going to track. This is only a small trial because we didn't have a lot of material collected. But the way we're going to look at scaling that up is to launch our citizen science program, which Lana's going to talk to you about next, but really get a lot of people on the ground and be able to collect larger amounts of material so that we can put it back in. We're also going to keep an eye for flowering. The New South Wales meadows are notoriously low seed producers, especially compared to the Posidonia in Western Australia. But we do see some flowering in New South Wales, so if there's an opportunity to collect seed and use it within local meadows as well, that would probably work really well once we've got some initial Posidonia growing as well. Obviously, we'll continue to monitor this site and just see how it's going to track. Posidonia is a slow-growing species, and so we won't even expect to see new shoot growth probably for 6 to 12 months. So they're kind of sitting there, but they're surviving at the moment. So, as far as scaling this up to estuary level or multiple estuaries, we really can't hope to do that unless we can really change people's attitude towards seagrass, towards their local, the way they use the water, particularly for boat owners. If we can't change block and chain moorings to environmentally friendly moorings, there's really no point going in and doing this work. So we're trying to find a way to actually get a whole estuary community on board and really scale this up as a proof of concept that we can recover these seagrass meadows, but everybody needs to be on board. There's no point trying to restore single mooring scars where EFMs have been installed when you've got a lot of block and chain moorings around. Sorry, finished. And this is, it's a collaboration between West Coast and East Coast brought together through the Seagrass Restoration Network. You want me to sit down? Lana's going to come and talk to us now. Lana's also working on Operation Posidonia, but she's going to talk to us about how we're going to engage our local community effectively. Thanks. Let's pull this up. One sec. Oh, sorry. That was my fault. Head down to there. Awesome. Thanks. There we go.
G'day everyone, my name is Lana Kajlich, and in a nice segue from Liz's talk, I'm going to talk to you today about the social research aspect of the same project, the exact same project, the project to restore Posidonia australis to the old mooring scars in Port Stephens. Basically, there's two big furphies in this slide. The title itself, it's not actually to do with seeds in terms of the transplants, so the transplants instead of seeds, and also the photo itself, that's actually from Western Australia, those beautiful flowers, not from the East Coast. Evidently, they're more beautiful, but I'll wait and see. Basically, today I'm going to talk about, give you some background on an old new approach in social research, old because it's been around for ages, and new because it seems to be everywhere that I look at the moment, called social network analysis. And I'm also going to give you some preliminary results from a pilot study that I'm ongoing collecting data at the moment. Everyone here has sort of witnessed how important people are in the restoration process. And like Bruce Clarkson said yesterday, you know, bring a politician along to active people restoring in the field and then see if the funding is cut the next year. So I'd just like to show you an example when it's worked really well, and I encourage you to go to Alex Campbell's talk at 3 o'clock as well, which to showcase the work of Operation Crayweed, which is the project to restore dominant seaweed to the coastline of Sydney. And basically, one of the principal researchers, Alex Campbell, who's in the room today, and we'll be talking later, it's involved not just governmental funding, but also private funding in the terms of crowdfunding and also NGOs. And that crowdfunding campaign attracted $40,000 in a matter of days, so it's pretty a testament to the power of networks and people. It also involved artists, approached scientists, and held a Operation Crayweed homage to seaweed at Sculpture by the Sea. And the City Council member came along to cut the ribbon and get involved. So that's just an example of when it's worked really well. And the benefits to the community and the contributions made by the community, not enough research has really gone into looking at those two aspects, but it would be great. So the new project in Port Stephens called Healing Old Ruins. Basically, it's a collaboration again, so we've got the expertise and the methods from the UWA and from SIMS, Sydney Institute of Marine Science, UNSW, but also the local DPI, whose headquarters of fisheries in New South Wales is based in Port Stephens, which is just a bit of the social ecology of, the social landscape of Port Stephens. It's located 2 hours from Sydney, but it's also got the Worimi Indigenous group on site. Head is Nelson Bay. It's got a long history of agriculture, of oyster farming, which you might have heard from Ian McLeod yesterday, and some of the oldest in the world, and it was at one stage the largest in the world in the late 1940s, and very low agricultural pressure as well. I mean, there's this fixable problem that's been identified with the traditional moorings in place at the moment, the sort of dragging chain against the seagrass. There's a simple solution which has been in place in places like Sweden and New Zealand and Tasmania for decades now, and they're called environmentally friendly moorings. And Moreton Bay, thank you, with those successful restoration trials, the successful trials that turned into being subsidised by the government as well. Fantastic. And legislated by the government as well. Very helpful. So, like we were talking about yesterday in yesterday's session, with the coastal scaling up session, we should be talking about mitigation of threats very much. And the question was asked, what can the community do at an individual level to mitigate those threats? And I hope that this can become a bit of a poster kid for a mitigation of threats, because that's a primary, absolutely tantamount importance, and should be taking place prior to restoration, or at the very least concurrently. So the mooring groups, this is one huge group, and I remember when I went to Manly in my local council, and I sort of was informed first time about EFMs, and that was something that, I called my parents straight away and let them know. It's just that awareness that EFMs exist is really important as a first stage. And then building trust in the product itself. And secondly, the local dog walkers and walkers are the first on the beach in the mornings, so they can be collecting the washed up shoots that come after storm events, so that it's not sitting there all day long and being degraded. So mobilising this group and encouraging and inspiring and involving them would be fantastic. So, and that's a great way for them to get involved. So those shoots would be either put inside their house and then later collected by DPI. And there's an opportunity there for interns and, employment as well, potentially down the track. So there's a science communication campaign, and I hate that word actually, but maybe outreach is better. It's a bit one-sided and one-directional. Called Operation Posidonia. In a couple of weeks, we're just about to launch it with the whole team of John Statton, Liz Sinclair, and the rest of the team. If we've made some really wonderful advances in science outreach in recent times, and if you're interested in a good paper, this Farmer paper has shown the way we're sort of exchanging knowledge and information between the community these days. There's an opportunity there as well to do some side experiments with messaging as well, looking at diversity of evidence and presenting it to the public. But basically, this is a serious work, but we can have some fun with the engagement side of it. The dog walkers can be mobilised and encouraged to join seagrass storm squads. And then there'll also be beanies, which sort of give a sense of belonging and attachment to the project. The mooring owners will be encouraged to look after their bottoms, and with the potential for monitoring below their boats using BROV techniques, and perhaps even displayed at the marinas to show what is actually under their bottom of their boats. And the research aims, the social research aims, are really to understand the baseline knowledge of attitudes of both seagrass and the EFMs, the environmentally friendly moorings. As a first starting point, we just need to know how we're going to design a science communication campaign and address all the questions that the community have and the gaps in the knowledge. And secondly, to identify who are we going to target to extend the reach. I mean, ideally, the messaging should come from the community themselves because they're the owners of the solution. And the timeline looks like this. At the moment, in about a couple of weeks, we're going to launch a campaign in Port Stephens. And just looking back a bit, I've conducted all these sort of extensive, very long interviews with the community, about 3 hours long each one with 50 people. And in September, and just the whole reason for that was, number one, to meet the community and find out who's out there, and but also it obtained social license as well. We brought everyone in early and let them know what was happening, and then developing the science communication. And the second part is what I'm going to talk about today, which is collecting pre and post science communication data. Looking at the two aims, how can we do it? The literature sort of suggests that we really need to acknowledge the role that humans actually play in influencing each other. So using that network approach, what you see there is often used in the public health literature in terms of you go in with a public health message and in an intervention they call it, and then you look at pre and post and see how extensively that message was taken for health campaigns. The circles in the middle are the nodes of the people, and the ties between them are essentially the relationships. And in this one, this picture, this sort of network graph by Valenci, it maps kind of well to our context in Port Stephens. We've got the 4 key stakeholders, which is City Council, Estuary Group, which meet regularly on seagrass issues, general public, and then the Yacht Club. And those relationships are who recommends who on seagrass issues. So who would you talk to Bob, he's an expert, and talk to John. And so that's actually showing, this is actually just a representation, not exact, of those relationships. And the central tenet of social network analysis is essentially the structure. The position where you sit inside that network is actually determines that it can constrain information flows or it can also promote it. So your position in that network is actually quite key. And this technique has been used in policy mapping at the moment. It's been used in physics. It's been used in a wide range of social networks, social research as well. So I went out as first course and interviewed the estuary committee of 30 people, purpose for snowball sampling, and it wound up being 50 interviews. And that means not surveys, but sitting down with people for about 4 hours, going to their home, grandkid on the lap, and you really chat about seagrass issues and mooring, if you know what they know about moorings. And you build a lot of trust and you build a relationship in that time, and it's a long time, and it's fantastic. The three things, this is like these hours and hours of interviews, but this is just a cursory thing. It answered 3 questions for us as well, like how in the past has the community acted on environmental issues and collective action? How are they getting their knowledge spread across small town, word of mouth? And the third thing, what's going to happen when a group of outside scientists come into this and give them some information? So there is a distrust of authority there in the Port Stephens, but hopefully by coming in early and building a relationship with real people helps a lot. Just to give a quick example and to stop there. Oh, that's great. There's a really good example of how this technique has been used in the culture of farmers adapting to climate change. Now, farmers are, they're obviously very risk averse, and a high percentage of agriculture practices do fail, and it's absolutely tragic. So, all the research is showing that with climate change, we need to move to more transformational, broad-scale, enormous changes. So things like moving properties to different locations, changing business structures. So we're really just making often incremental changes, and we can understand why, because it's a risky endeavour. But this study looked at the role of the networks, and that dot in the middle is an individual farmer or representative, and the lines are showing all the social ties, all the close friendships ties that they have to someone. And that's actually a total, it's not an average of the 29 farmers. And they looked at people who are doing incremental small changes and they found that they had a really dense social network. And that sounds naff to talk about social social groups in terms of serious farming changes. But if you think about professional life, how much social support has helped with you making different changes, etc. The transformational changes, you can see, have the opposite. They say, I prefer to go it alone. If we look at the information network, where they source their information, those transformational adapters down the bottom, essentially the picture is really reversed, and you've got them seeking actively knowledge about how to make those changes. And whereas the ones with the incremental are like fewer sources. So the parallels to the marine context, like we've got this mixed marine use, mixed diverse space, but the central question is the same: who are we going to target, and who can we also include on the peripheral? Like, how can we include people typically not included in these campaigns? So all this cognitive psych literature sort of points to the individual attributes. So that's great for telling the science communication. It's still important, but the role of social networks also need to be pulled into it. Look at the group as a whole. So in that group, I'm looking at individual factors. And I'm just testing whether we can categorise these groups and look at who are these people who are going to best spread the information. Is it the innovators who are quick to adopt new technology, in the case of EFMs? Or is it the greenies, the ones who sort of self-ascribe to being quite, green? So we'll categorise. And the second thing I'll look at is the same as the agricultural paper. Look at the social factors and the role of that social capital that's go through. Sorry. No, I'm not sure which button. There we go. That's great. Just to touch really briefly on the categorisation, because it's super important. It's part of a broader research that I'm looking at in environmental stewardship, and I'm with a collaborator. I'm looking at from Port Stephens down to Jervis Bay, and sort of like looking at all the categorising types of behaviours that high Uber stewards might partake in, and the sort of the value that might have in terms of ecological values as well. And in terms of categorising how, the techiness they are and the level of innovation, I'm using from Jackie Moore's plenary speak as well, this Rogers innovation scale. And just looking at the innovators and the early adapters. And the dogma is that, you try to convince them to the innovators and these early adapters first. You don't look at all the laggards, etc. But, of course, in a campaign, you do want to include everyone in the picture. But this is for obtaining critical mass and spreading the message effectively, efficiently and quickly. So just some results from the pilot. It's a sample size of 13. Went down to the mooring club, the yacht club, brought cheese and not coffee, but chocolate, and had a chat before the guys had their beers. And then in a subsequent session, I sat down with them one-on-one and asked these questions about the social stuff, because that's kind of personal. They scored low on environmental stewardship, although the yachties said that they're really, really green, so they perceive themselves, but really differently on that, diffusion of innovation scale. I just want to show you, this is an aggregate of the 13 mooring owners. 4. That's wrong. 4 of them actually ascribed to being very eager to try new adopters, so they'd be put in the early adopter stage. And 8 of them said unmotivated by advertising, and I'll buy it when it's almost outdated. And what that showed was we looked at the source of information, where people got to. These early adapters, they went actively. They were going to the 3 mooring clubs that they went to. They were going to Sydney, and it's specific about finding information about boating equipment. It's, have to be really specific, not just generally in life. This guy, these 8 people, it was had just one source, and it was a local mooring contractor. It was very important. The share networks, that's who they share the information with, very similar. But the support networks, that's the social support. How many friends would you say support you in your decisions in, boating, buying big boat stuff? And here we go, the innovator, or the adopter up the top, has just, I prefer quality over quantity. And this person was saying, I've got lots of mates, cousins, friends. Sorry, that's an aggregate, actually. So that's how it looks. It was very different, that structure. So the next steps are actually to look at the early adopters and categorise them. It is okay. Thank you. Sorry. And looking at pre and post science communication and tracking that flow of knowledge, attitudes and actions, whether they adopt or inquire about EFMs. I'm sorry about that, ran out of time. Yeah, and then, yeah, thank you so much. Cheers. Oh, sorry.
Okay, John, you've got 10 minutes. It's my fault. So last speaker is John, and he's going to talk to us about some seed restoration work, which we're really trying to scale up in Western Australia.
Thank you, Liz. Okay. Do this quickly. Okay, I'll fly through this as quick as I can. Okay, so title's developing the steps required for effective use of seeds in seagrass restoration. So locally and also globally, we are trying to collectively develop up methodologies and techniques to actually utilise seeds for seagrass restoration with the sole purpose of being able to scale up our restoration. So you've heard a little bit about from Jason regarding the facilitation method with Amphibolis Antarctica seedlings. Quite promising. Unfortunately, there was a little bit of disturbance, which caused some problems there, but it's still quite a promising approach. In the US, we have these large numbers of seeds collected. They've been at the game a lot longer than we have here in Australia in terms of this process. We've been able to collect large numbers of seeds. In South Australia, they've also been using newer techniques for seagrasses, but old techniques for terrestrial restoration, removing topsoil containing viable seeds, and that's been quite successful in terms of scaling. More recently, we've been playing around with a few different species in Western Australia, in particular Posidonia australis and also Holophila ovalis. Now, Western Australia is a biodiversity hotspot for seagrasses. We've got about a quarter of the world's seagrass species occurring along our coastline. We have a very large coastline, of course. And so that means we have a lot of different species that we could potentially be tapping into for restoration programs. So in saying that, there's a large diversity of form and function with seagrasses. We have these large climax persistent species here on the left-hand side, and then we've got these sort of faster growing colonising species here on the right. And these larger species are quite tall, up to 1 metre height, can reach up to 2 metres in Shark Bay. These are about 50 centimetres, and these are only centimetres off the sea floor, and up to 10 centimetres here. We also have a diversity of form amongst the early life history stages. So as Jason showed, Amphibolis Antarctica has these grappling hook here used to attach to fibrous structures. Posidonia australis has this large endosperm, and we have these other two species here which have these small hard-seeded, seed-coated seeds. And we've been able to conveniently, previous research back in the 80s has been able to conveniently categorise these into 3 different groups. So we've got no seed coat, no dormancy, growth attached to the parent, so they're viviparous. With this fleshy seed coat, no dormancy, so these are direct developing species, and there's several of those. And then we have these hard seed coat with a distinct seed dormancy. So about 90% of the seagrass species have a hard seed coat and some level of dormancy. So how are we trying to improve seed-based capability? So we're doing this through adapting some terrestrial-based approaches. I previously used to work up at Kings Park with Kingsley Dixon and the team up there some years ago, and so I'm still collaborating with those guys today. And so some of the things that we're looking at doing is how do we source, or where do we source, and how do we collect the large quantities of seed required for restoration? How do we ensure that it's processed, stored and managed to ensure that these seedlings are high quality? How do we deliver these seeds to sites so that we're actually delivering highly viable and quality seeds? And then also this more recent research is what are some of the ecological barriers to these seedlings actually establishing? So can we identify these and potentially overcome them? So just to go through quickly, we've been developing some capability in terms of understanding where and when to collect these different species. So in Western Australia, Amphibolis Antarctica, we're seeing that it's readily available in May, June around the metropolitan and southern coastlines, and then it gets pushed up into about August each year as you move further north. Similarly, we have a latitudinal gradient for Posidonia australis, where we have this differing availability in fruit. They're only available at one time of year, and we're able to track when these fruit are available. Holophila ovalis, in the Midwest, it's available all year round. If you move further south, it's available around January to May. and then it tends to only be available in the dry season in the northwest, mostly because of the weather conditions in those particular locations. So some of the ways that we can collect the seed, as Jason mentioned, we can facilitate seedling recruitment using these Hessian bags. It's been quite a successful method. Fruit of Posidonia australis can be collected as drift in the water column or directly from the parent plant, or even as beach cast as well. So this is a 300 metre long beach and this is 2 metres wide. It's about 10 centimetres deep of fruit and fruit with seed as well, so huge numbers there. In the US, they've been able to automate this process using this little contraption that sits on the front of the boat with a cutter head here, and they're able to collect the flat reproductive structures in that instance. And more recently, we've been playing around with using an otter trawl to collect Posidonia australis fruit. It's been relatively successful in collecting the fruit. We've also been collecting a bit of bycatch as well, a few scorpion fish, crabs and some other little critters in there as well. So it works quite effectively. This mouth here is about 2 metres wide, and we can tow this at this speed. We can cover some quite large areas. Once that fruit has been collected, it can be processed in a number of different ways depending on the species. So Zostera marina, they tend to leave it in tanks to sit over a number of weeks. The spathes, the reproductive structures, split open and the seeds fall to the bottom of the tank. Posidonia australis, we use a similar method, but we actually increase the speed of this by agitating the fruit in a tank system and also keeping it at a certain temperature in a climate-controlled facility. Let's see if this will work. And the objective of this, sorry about the noise. So the objective of this is to actually obtain pure seed. So we don't want to have all this husks of the reproductive structures and the fruit left behind because it ends up contaminating the seed. So Zostera marina, they get this pure seed at the bottom here. It goes through a small filter and we get seed. Posidonia australis, we collect it on the bottom of these tanks here. Again, the fruit husk and the seed separates because that floats and that sinks. And then Holoflora ovalis, we have the same process where the seed separates out through a mesh screen and the seed settles here on the bottom. So we're able to contain, this is only a pilot scale, we're able to contain large quantities of Holoflora ovalis seeds using this method. Quality control of these seeds, so Posidonia australis, the great thing about that, Posidonia australis, is that the seeds, you can already see that it's viable, it's got a developing shoot and root system. However, the quality of the seed also needs to be checked, so we can section these seeds, stain them with potassium iodide, and if we see large amounts of starch in there, it's a good quality seed. If we're not seeing large amounts of starch, then we're seeing that's probably a poor quality seed. And also the size of the seed has a big impact as well. So seeds generally are around 15 to 18 millimetres in size. We do get some up to around 26 millimetres, which are great, but much smaller ones exist as well. Holoflora ovalis, the dormant seeded, the hard seed coat. We've tried tetrazolium staining, but it's not very clear when we've done this method. We've applied a squish test and a sinking test, so if they squish, they're not viable, and if they sink slowly compared to other seeds, it means they are less viable. And I have a student at the moment who's in the process of testing that out to see if these actually relate to the viability of these seeds. But this is the great thing about this particular method is that we can do this in seawater, so it doesn't actually impact, we're not destructively sampling the seed. We can actually use them back in the trials if need be. We've been looking at how we store and manage these seeds. So as I mentioned before, a large majority of seagrass species have a hard seed coat with a